My name is Michael East from the University of North Carolina, and today I'll talk about my efforts to functionally integrate understudied kinases into signaling networks using quantitative omics data. When we think about kinase biology, we tend to put kinases in the context of these linear signaling cascades like the EGFR signaling pathway shown here. But in reality, these linear kinase pathways don't occur in a vacuum, and signaling is much more complex. Shown here are just some of the KEGG pathways that EGFR contributes to, with kinases highlighted in red. As you can appreciate from these pathways, kinase signaling is incredibly complex, with a very high degree of crosstalk between canonical signaling pathways. When you consider that a third of the more than 500 kinases that make up the human kinome are considered dark kinases, understanding this complexity becomes a major challenge. We and others have also shown that in addition to being highly complex, the kinome is dynamic and very resistant to perturbation. And work from our lab, we've shown that inhibition of one of the kinases in this signaling cascade, MEK, results in only the transient decrease in activation of its downstream and substrate kinase, ERK. Within hours of MEK inhibition, we see upregulation of a number of other kinases, ultimately resulting in adaptive bypass of MEK inhibition and reactivation of ERK. When we use our MIB-MS technology to profile the kinome in response to MEK inhibition, we see sweeping changes to the kinome, including upregulation of a number of receptor tyrosine kinases, including DDR1, AXL, and PDGFR-beta. Now, in this example, the end result of kinome remodeling was the acquirement of drug resistance to the MEK inhibitor. We saw only a transient effect on growth in response to MEK inhibition in these cells. In other studies, other labs have shown that kinome reprogramming contributes to plasticity in the cancer stem cell phenotype, as well as to epithelial and mesenchymal state changes. So kinome remodeling or reprogramming has very real, very clinically relevant consequences on the biology of the cell. The goal of this project is to leverage the dynamic nature of the kinome to better understand kinome complexity and to integrate understudied dark kinases with well-characterized kinases into functional subnetworks that drive kinase biology. To accomplish this, we've taken an omics approach, combining RNA-seq to measure transcriptional changes and MIB-PRM-MS, as described by Robert in the previous talk, to measure changes in the functional kinome at the protein level, to study the consequences of a diverse panel of cellular perturbations, shown here on the left. We then used computer modeling to define kinase signaling subnetworks based on how the kinases respond to these perturbations. For the purposes of this talk, I'll focus only on our RNA-seq data so far. Shown here are the effects on kinase expression for each of the different inhibitors tested in a breast cancer cell line SUM159. These cells were treated at IC90 concentrations for 24 hours. In these kinome trees, red circles indicate upregulation and blue circles downregulation of their respective kinases. The size of the circle correlates with the magnitude of change in message abundance and ranges from a 2-fold change all the way up to a 64-fold change in expression level. Even though all these perturbations had a fairly robust effect on cell growth, the response of the kinome was unique to each perturbation, as these kinome trees are visually very distinct from one another. When we performed a correlation analysis, we saw that kinome reprogramming is very divergent across these perturbations. The 3 kinase inhibitors, palbociclib, MK2206, and trametinib, were the most similar, with R values of about 0.6, but the kinome response to the other perturbations showed even lower to virtually no correlation. If we consider all the kinases that respond dynamically across all perturbations indicated by the circles on this kinome tree, we see that approximately 350 kinases respond, which equates to about 75% of the expressed kinome in this cell line. This percentage is consistent for both the well-characterized kinases, shown here in pink, and the understudied kinases, shown in green. Considering only the understudied kinases, about 95 of them are expressed in this cell line, and about 70 of these kinases are dynamically regulated by at least one perturbation in the panel. This shows that the expression of almost every kinase can be regulated dynamically in response to changing conditions within the cell. I think this finding is very important, as we've only really recently come to appreciate the dynamic nature of the kinome. Traditionally, we thought kinase signaling was regulated primarily by post-translational modification at the protein level, and that kinases were otherwise stably expressed, similar to housekeeping genes. The scope of kinome remodeling shown here further illustrates the complexity of kinase signaling in the cell, and taken together, these data demonstrate a diverse and extensive response of the kinome to perturbation, which is critical for our computer modeling efforts to define functional kinase subnetworks. And by putting kinome reprogramming back into the context of genome-wide transcriptional changes, we've begun to define functional gene clusters from this data set. In addition to all the changes to the kinome, these perturbations also result in robust transcriptional remodeling with thousands of differentially expressed genes, summarized in this table. Now, we're still refining these models, but we've begun to identify responsive gene clusters that are heavily enriched in different gene ontology terms, indicative of a common biological function. Shown here are two such examples of these functional gene clusters. The lines in these graphs represent kinases within these clusters, and the green lines represent understudied kinases, which are also identified on the right. Although we have not directly tested kinase involvement in these biological functions, such as cell cycle for cluster 4 and lysosome and autophagy for cluster 10, many of these kinases have functions consistent with these Go terms. For example, cluster 4, which is one of two cell cycle clusters we've identified, contains TLK1 and TLK2, which regulate DNA synthesis and chromatin assembly, and NEC1, which regulates the DNA damage checkpoint. This is in addition to other well-known, well-characterized kinases that regulate the cell cycle, including aurora kinase B, BUB1, CDK1, CDK2, CHEK1, and WEE1. Now, while we're still building and refining and validating these gene clusters and kinase subnetworks, we've demonstrated the capacity of the kinome to respond dynamically to perturbation, and are defining functional kinase subnetworks that place understudied kinases in the context of other better characterized kinases involved in canonical signaling and biological pathways. I'd like to thank other members of our lab and our collaborators both here at UNC and WashU, especially Sean Gomez, Chinmaya Joysa, and Matthew Brzezinski here at UNC, and Reed Townsend and Robert Sprung at WashU. And with that, I'd be happy to take any questions. Thank you.